colleagues, ladies and gentlemen, I'm very proud and happy to start today's Brownbag-Session here at the UNESCO Chair for the Management of Conservation Areas in Carinthia at Carinthia University of Applied Sciences. Today, we are happy to welcome Ana Rodriguez Prieto from the company WonderGene. Ana is the managing director of a small startup company that addresses barcoding and environmental DNA issues. We are very happy, Ana, that you can be with us today. And we will ask you this question: how can mobile labs function in a muddy jungle and ensure wildlife conservation? Ana, the floor is yours.
Thank you very much for the presentation, introduction, and hello everyone. So I'm gonna share my screen. Okay.
It's perfect.
So, perfect. So yeah, I would like to start with a brief introduction of myself. So as you can see, I'm a biologist, but also a biochemistry. I study also biochemistry, just because I love nature, environments, animal, plants, and all the living organisms that lives in nature, but I also love lab. So I decided to do this double degree just because I was interested in two things. Then I moved to the south of Spain to do my master degree in the National Park of Doñana, and then I moved again to Barcelona to do my PhD in evolutionary biology. Then on 2013, I moved to Trento here, where I'm now today, in Italy, to do my postdoc. The main focus was integrative taxonomy, and then I'm gonna explain you it's better. Now I'm the co-founder and CEO of WonderGene. That is an innovative startup. So today my presentation will be focused on my research project that I developed here in Trento. I came here in Trento to work in the Muse, that is the most innovative science museum in Italy. And I came here to work with the Department of Biodiversity Conservation in tropical environmentals. The researchers working in this group, they were working to study biodiversity in this ecosystem that is called Afro-Montane region, that you have here a map. You can see where they are. This Afro-Montane region is a group of mountains in Africa, and they are really particular because they work like island. I mean, they are not connected between them, so the biodiversity in them is really special and particularly. Our research was focused mostly on amphibians and reptiles, and the idea was to make a species identification, a species delimitation, always with a focus of wildlife conservation. So one of the first things that I have to do when I arrived to the museum was to set up a genetic lab, because in the museum there was not a previous lab. So here you can see a photo. It was a little room, but it was really nice to be able to set this lab. So as I told you before, all the research was focused to do studies and research doing integrative taxonomy. That means that we combine morphological studies, ecological studies with genetic information. So talking about genetic analysis, my part was in this project was to make barcode analysis. I'm gonna briefly explain to you if you don't know what's the barcode analysis. So barcode analysis is a kind of genetic analysis that we use to identify species. So analyzing a concrete part of the genome of animals, plants, bacteria, virus, fungi, whatever organisms, we can identify the ID of the species. Normally, when we made the barcode analysis, we have to follow a simple procedure that you have represented here in this scheme. I mean, we have to start with DNA extraction. This is the first step. And I put in here also the equipment that we need, because it's going to be important for the next steps of my talk. So here you can see that we use normally a centrifuge that is like equipment that's a little bit big and really heavy one. The second step, after the extraction, you have to amplify the DNA, but we are not amplifying all the genome of animals or plants that we are analyzing, but we normally amplified the barcode marker that for animals is COI1, for plants there are 2 markers, fungi have others, and so on. After this amplification, you need to read the DNA that you have already amplified. I mean, you have to sequence in. So normally we use sequencer as the one that you can see here in the slide. They are huge equipment. Finally, when you obtain your sequence, you can use it you can check it against a public genetic database, and you got a result. If in this database is the species that you are analyzing is already there, you are going to have a match. If not, you are in front of 2 different situations. Probably you are analyzing a new species, or probably no one before you have analyzed the barcode of a already known species. Okay, so this is a normal procedure that we have done in the lab, and we have done also here. I mean, we have done in our research project. So I'm putting here this map here because in the pin in the upper part of the map you can see where the Museum of Trento is, and in Tanzania you can see the point where the Muse has a field station in the middle of the Udzungwa National Park. So if you would like to go from Muse to this field station, you're probably going to take something like 2.5 days traveling, plane, bus, car, whatever. But the problem is, if you want to bring animals, plants, or whatever you have collect in the field directly in Tanzania, and you would like to bring this specimen back to the museum to analyze them morphologically or genetically, probably they are going to take something like 1 year or 2 years to arrive. Why this? Just because of bureaucratic things. I mean, if you would like to do research, as always in our countries, you need research permits, and this can take a long time to get them. Also, you need export-import permits, and with customs, the situation of moving specimens is not so easy, obviously for normal reasons related with CITES and also with endangered species and diseases. But the thing is that if you would like to develop studies to study biodiversity in a place like Tanzania, probably it's going to be hard to get the results on time. Why I'm meaning on time? Because if you are study, an endangered species, or if you would like to make a biodiversity assessment quickly, it's not going to be possible to do in the traditional way, meaning that you go to the field, collect samples, and then you would like to bring back them with you to a museum here in Europe. Probably you are going to be late. So for this reason, the second request of my postdoc was, it is possible to make a genetic analysis directly in the field. So the first thing that I made when my IP asked me to this, I started to search for information about biodiversity distribution and sequencing or molecular labs facility distribution in the world. Here you can see on your left a map of richness distribution of amphibians in the world. So probably in the tropics, they are more abundant. And then in the right part, you have a map where the sequencing facilities are in all the world. So probably there is a mismatch between the distribution of the concentration of biodiversity, in this case amphibians, and the distribution of sequencing facilities, so places where we can analyze by genetics these animals. I'm reporting here amphibians, but this map can be the same for all the terrestrial vertebrates. I mean, they are more abundant in the tropics. So here we have a problem. If we would like to develop a genetic analysis directly in the field to identify new species or to make a species delimitation, it's not so easy. So for this reason, we decided to start a project. The name of the project was Expedition Lab. So the idea of this project was to be able to perform a genetic analysis directly in the field in tropical forests in Africa. Here you are seeing a picture of the tropical greenhouse that the MUSE has. So I put it here because it was a fundamental part for developing my project, because I use it like a lab test for testing all the equipment and all the reagents that I have to bring with me in the field, in the reality. So I test them here because the environmental conditions are the same. So environmental condition, this is the most challenging things that I faced. So not so for the higher temperatures, because the temperature in these forests are something like 25, 27 degrees. So it's not so different from here. Probably here we are suffering now hotter temperatures, but the problem for sure was humidity. I mean, humidity here are something like 95%, 99%. So humidity for equipment is like a killer, because probably all of you have a personal computer, and a personal computer have fan to prevent overheating your machine. So when a fan gets wet, the equipment is going to stop. So one of the things that we have to work with was against humidity. Obviously, also a tropical forest. Comparing with a traditional lab, molecular lab, you don't have electricity. So most of the equipment needs electricity, and also most of the reagents need to be in the fridge or in the freezer. So yeah, this was the 3 parameters that we have to work with. Here I report all the partners that support this project. MUSE, obviously, Università di Verona, Fondazione Caritro that make a funding possibility, and Wildlife Conservation Society. And probably the most important part also was provided by Oxford Nanopore Technologies, that at that time in 2014, they were also in a startup, and they have developed a portable sequencer. So that was the most difficult thing to bring to the forest, a sequencer. So they have developed this, and we entered in the beta testing program. So they support us to prove their technology directly in the field. So here I report you a little bit in a scheme of the equipment that we bring with us in the forest. So for doing the DNA extraction, we find on the market a little thermocycle, that is the oven that we use to amplify DNA and to make the DNA extraction. The important thing is that this thermocycle works at 12 volt, meaning that you can charge it by portable batteries. And I make some modification to prevent that the humidity enters in the fans, and to make it work properly. Then the second photo represents the centrifuges that we developed by ourselves, because normal centrifuges, they were impossible to bring with us. It's like a washing machine. I mean, they weigh a lot because they have this heavy thing to prevent them to fly. So we decided that there wasn't any commercial solution that had the characteristic that we want to have. So we construct our little centrifuge with a motor of a drone, and some Arduino, and I mean, DIY fabrication at least. Then for DNA amplification and quantification, again, we use the same thermocycle. And for the quantification, Normally we use something like fluorimeters. Fluorimeters are really particular equipment that they have to be calibrated and they have to be really stable, because if not, they are not working. But they are not, I mean, good for bringing them to the forest. So we decided to build one for this purpose. And as you can see in the photo, we put the fluorimeter inside a metal box. That they are the boxes that you can see in our cities where the cables of electricity and telephone cables are. So we use it to prevent the humidity to enter in. Finally, as probably the most important part of our idea was the sequencer of Oxford Nanopore Technologies. So then we work a lot with reagents. I mean, we try to avoid them to be at temperatures from -20 to 4 degrees, that it's normal temperatures that you have to conserve or maintain them. So we lyophilized a lot of them, and we stress them in the tropical greenhouse to test them to adapt them to work in a tropical environment without any fridge. So here you can see the setup for the first expedition that we made in Tanzania in 2015. We went to that field station that the Muse has, like a base camp. And this expedition lasted only 10 days because it was the first trial. We like to test our idea. I mean, it was so challenging, so we were there to try it. This is the second field station that we made to the analysis. So it's something like from the field station of the Muse, that is like something like a little village. We made something like 4 hours driving and then something like 4 hours walking through the forest. Okay, so that was the camp with our tents, the kitchen, and everything. Here you can see some of the picture about us working. So yeah, we changed bench of our molecular lab for suitcases, tables, battery cables, muddy, and everything was really a big mess. But we're really happy because we reached our objective, that was to sequence one animal, one specimen. We sequenced a frog. So here you have a screenshot of the sequencing process. I mean, this is the output from Mini-ion. So we were really happy, and we published it in 2015. And probably was one of the first papers that were being published about doing or performing a genetic analysis from extraction, amplification, and sequencing directly in the field. So we were really happy. We were really happy, but we have a lot of things to improve. So for this reason, we worked for 2 more years, improving our equipment, improving the protocols, the reagents, all the things. And we decided to make the second expedition to Democratic Republic of Congo in 2017. And we spent there 1 month, so longer expedition. Precisely, we went to study biodiversity in the Kabobo Massif. That is incredible forest. And here you have a map. I mean, it's the largest stretch of mountain forest in Eastern DRC. It's about 100 kilometers long in the western shore of the Lake Tanganyika. And it's really interesting about biodiversity, because we probably think that this forest has been isolated in the last 10,000 years from the other forests. So the biodiversity there is incredible. And probably, as you are thinking now, I mean, this is not the easiest place in the world to go to make research, because a lot of problems about illegal mining, war, political situation. So not so many people has been there to study biodiversity. Probably conservation of biodiversity is the last problem of these people. But we were there because in this year, in 2017, it has been a change of a conservation decision And they would like to have a biodiversity assessment of this place in order to know how important is this place and how to protect this place, and which can be the measure that they have to put on to protect this area. So here again, you have some picture of me doing genetic analysis there directly. Here in the first one, you can see the Lake Tanganyika. I think it's the most incredible place where I made a genetic analysis in my life. The view was incredible. I mean, I was doing there, seeing all the Lake Tanganyika, that I think is amazing. As you can see, also a big mess because, I mean, we bring with us a lot of things. My table, my bench top was made of bamboo, that it was incredible. And I was doing the analysis under a big mango tree. So incredible situation. And then we were really happy also because here I'm with a beer, really happy with a big smile, because we be able to sequence 2 mammal species, 2 reptile species, and 3 amphibian species. Probably the 3 amphibians are new to science, and also reptiles. So not because we are the best one, but probably because no one have gone there before and performed a genetic analysis there. So yeah, I think this is really important, because doing a genetic analysis directly there can give you a quick view about biodiversity. It's the first signal that you are facing a really interesting place. So in this way, you can give really quick answer to stakeholders that they have to decide what to do with this place, how to protect this place, and if we are in a hurry to protect this. So as all the research projects finished, unfortunately, with the great results of our research, in 2018, I decided to found WonderGene. As I told you before, it's an innovative startup. And the big idea of WonderGene is to aim to perform genetic analysis outside of a traditional lab, where these genetic analyses are needed. I mean, like I told you now, in a forest, because we like to make a biodiversity assessment quickly, or maybe like we are doing now, that we are giving our system to food supplying companies to combat the food fraud. So the idea of WonderGene is to provide these companies an integrated lab for DNA extraction, amplification, and quantification. So all the things that you have done, you have seen, so the thermocycle, the centrifuge, the fluorimeter, the pipettes, all the tubes and the reagents, we compact it in an integrated lab. And also we are doing custom and ready-to-use kits to give them to control by barcoding analysis the ingredients in our foods. And maybe you are thinking about, but what is the relationship between biodiversity conservation and food fraud? So here I bring to you a paper from 2022, so this year, as a paper, I think on March has been published, that is a study of researchers in Singapore that have analyzed pet food by barcoding analysis, and they have identified many endangered shark species inside this pet food. Here you have a list, a graph, where you can see the species that they have found in this pet food, and most of them are in risk. I mean, they are endangered by the USN categories. So I think it's really important to connect that most of the food that we normally eat have a relationship between biodiversity conservation, because we are eating animals, plants that can be endangered. So providing the technology and the ability to perform this genetic analysis to this food supplying company, I think can make the difference to biodiversity conservation. I would like to finish my talk with this images, these maps represent the biological field station that are now in our world. In the A picture, you can see biological field station that work with terrestrial ecosystems, the B, water ecosystem, the C, freshwater ecosystem, and the D, several kind of research. So I will really like that in the few years, some of these field stations has, like the green points, our portable system, and they also be able to perform a genetic analysis directly in the field really quickly and to get results that probably I think they are going to be important to biodiversity conservation. So thank you very much for listening to me. I'm here for your questions.
Will ensure nature conservation in the 21st century. Be part of the new 4-semester part-time master program. Carinthia University of Applied Sciences.